All right, so we're very excited today to have Dr. Jason Collier coming from Pennington Biomedical in Louisiana State University and Dr. Clive Wasserfall from the University of Florida. Both of them kind of in the midst of some stormy weather down there in the South, so hoping that all these connections will remain intact. The paper that just came out was really exciting, and the title of their talk is "Do Immunometabolic Events Contribute to Partial Remission in T1D?" A little bio for both of them. Jason Collier is an associate professor and director of the Islet Biology Lab at Pennington Biomedical Research Center in Baton Rouge. And the Collier Lab studies cytokine signaling in pancreatic islets with a focus on physiology and pathophysiology of such signaling events in pancreatic beta cells. Jason has a long-standing interest in immune cell infiltration into pancreatic islets and the action of cytokine receptor signaling pathways. Collier Lab uses mouse genetics and functional genomics to understand signaling pathway paradigms that are important for islet remodeling and contribute to disease-related events. And for Dr. Wasserfall, Clive Wasserfall trained in clinical laboratory medicine in South Africa prior to pursuing graduate studies in the US and Germany, and his laboratory focuses on the immunopathogenesis of T1D. Aspects of the work include refining prediction of disease onset and progression through the use of genetic, serologic, and autoantibody assessment. The mechanism of disease is explored on the cellular molecular level to better understand the pathogenesis of this disease and thereby lead to the goal of their program, which is to prevent T1D at risk. Their laboratory also houses the autoantibody screening core for the JDRF, or Breakthrough Network for Pancreatic Organ Donors with Diabetes, or the NPOG program. I'm going to drop the title and link to the paper now if anybody wants to take a look at it while we're in the talk. And thanks, both of you, for joining us, and please take it away.
All right. Thanks, Monica. Thanks for having Clive and I on. We appreciate the curated content that the Sugar Science puts out. So we're excited to be part of this platform. I just want to say about the title, we purposefully put a question mark here because we are not here to say we have definitive answers or we've solved all the problems. We're just putting forth what we think is a logical notion that may help explain some of these concepts. And we hope that that facilitates and continues this discussion. What I thought I'd do is start with the disclosures. I'll speak for both me and Dr. Wasserfall when I say that we have nothing that needs to be disclosed that would affect the impartiality or objectivity of any of the content you're going to see herein. The next thing I'll say is here's our real disclosures, which are we are not intending to provide clinical practice recommendations. We are simply putting forth a concept that we think is rooted in strong preclinical data in many cases, in some cases clinical data where it's available. Our content is based on published data, but I wanted to be clear that some of that is, and in fact, a large part of it is not exclusively using T1D models. So some of these are in other diseases, or they're just standard mouse models where a metabolic concept or an immune cell event or a beta cell event was studied in a non-T1D context. But we think transitioning from normal glycemia to hyperglycemia and using insulin to restore glycemia may be important in the context of some of these metabolic events. And then finally, Dr. Wasserfall and I agreed that we wanted to point out that there's an established heterogeneity in autoimmune diseases, and that includes T1D. So in many cases, things we say, we don't mean it to be a one-size-fits-all paradigm. We understand that across tissues, across individuals, and we'll show in some cases, even within a single individual, there can be heterogeneity depending on what parameter that you're measuring at that time. So we wanted to be clear about that. All right, so just a quick outline. I think most people that tune into the Sugar Science have an interest in T1D, so we're not going to belabor the point of T1D onset, but we do think it was important to have a brief introduction, particularly the staging of the disease. And I think the formalization of those disease stages came out about 10 years ago and has been, in my view, and I think Dr. Wasserfall would agree, a benefit to the field to divide the disease into stages. We're going to discuss partial remission and how there's certainly heterogeneity there, but there's also no formal definition. I'll say one accepted definition that everyone uses for partial remission. And then we're going to talk about briefly the influences of hyperglycemia directly on the islet beta cell. And then also how hyperglycemia may provide sufficient substrate usage for T cells and macrophages that essentially drives an inflammatory process, and potentially how that could be dampened or reversed with insulin therapy. All right, so just briefly, anyone who doesn't think about pancreatic tissue on a regular basis, particularly in the context of just normal physiology, here's the anatomical location of a pancreas. And I wanted to say that it's a very interesting organ because it's got two distinct functions. About 98% of the tissue is responsible for the production of digestive enzymes and associated ductwork to get those enzymes where they need to be. The remaining 1 to 2% of the pancreatic tissue are responsible for protein hormone production. And this is in specialized structures called the islets of Langerhans. And their job is to synthesize and secrete protein hormones that control the storage and utilization of simple metabolic fuels like carbohydrates, proteins, fats, and also regulate other processes. And so if we move this briefly from a cartoon to actual pictures of human pancreatic tissue, this is from Susan Bonner-Weir's group at Harvard. You can see exocrine tissue here on the outskirts of the image. And what this shows is two different islets stained for insulin in the red color and glucagon in the green color. And I just wanted to use this quickly to show that in most mammalian cell types, the insulin-producing beta cell is the predominant cell type. So many of us who use mouse or rat models, preclinical models in our work, that's certainly true. And also in human pancreatic islets, the insulin-producing beta cell is the predominant endocrine cell type. All right, now what's exciting and interesting about the pancreatic beta cell is the secretion of insulin is coupled to the metabolism of glucose. All right, so glucose is a 6-carbon sugar, and that's why you see this hexose shape here. It's transported in through a high Km glucose transporter, phosphorylated once it enters the cell by a high Km hexokinase. And that's important because those high Km transporter and kinase allows the beta cell to function as an exquisite glucose sensing cell. All right, it metabolizes the bulk of its glucose through glycolysis, which is 10 reactions that essentially promote a net oxidation and shorten that carbon chain to 3. And that pyruvate can enter into the mitochondria. The pyruvate can be converted to acetyl-CoA and enter into the TCA cycle, which is another 8 reactions that essentially promote oxidation reduction energy that can be coupled to phosphate bond energy, right? And it's important because those ATP that are produced by 1 mole of glucose, you typically get 30 or 32 ATP, depending on whether glycerol phosphate or methylated aspartate shuttle are used. But regardless, the accumulation of that cellular ATP will close an ATP-gated potassium channel. And this triggers a depolarization event. The beta cell essentially undergoes an action potential, just like analogously to a neuron, opens a voltage-dependent calcium channel, and both extracellular calcium stores or signals and intracellular calcium signals are important for the trafficking and exocytosis of those packaged insulin granules. And so it's a very elegant system of coupling glucose sensing to insulin output. Very subtle raises or lowering of blood glucose. I'm going to show that briefly in data form using the rat. So this is an older paper by Unger and Orci. And what you're seeing here is glucose level at 60 mg per deciliter. Insulin output is at a baseline level. When that gets raised to 80 mg per deciliter, which is a very seemingly subtle shift in glucose in the blood, beta cells are able to sense that and respond with an insulin output. Those beta cells essentially reset. You can see that glucose has dropped to 70, and then they're reinitiated with a stimulus at 90 mg per deciliter. Again, not a very high level of glucose, but still promotes an insulin secretion event through that coupled glucose metabolism. Now, at the same time, the alpha cell responsible for glucagon production has a higher baseline level at low glucose. You can see opposite to insulin. And the response of raising glucose triggers an insulin output. And what you see is a lowering of glucagon. And so the insulin-glucagon ratios kept in harmonious balance are important for overall glucose homeostasis. And I think that's reflected really well in continuous glucose monitoring over time. And so what you see one individual, black arrows indicating meal times, gray arrows indicating intense exercise. And what you can see across a large period of time, very, very strong control of glucose homeostasis, afforded by the minute-to-minute control of intact pancreatic islet endocrine cells. Contrast that with an individual that has T1D. And what you see is, despite this individual being reported to have good glucose control with their injected insulin, a lot more glycemic volatility, right? And this, I think, really showcases the difference between an intact functioning beta cell versus an injected insulin. All right, so if we were to move on to talking about T1D and how does T1D develop, I think most of us agree that there's no one accepted trigger for the disease. It can be very heterogeneous onset in terms of age. And one of the things that we alluded to earlier is how the field divided it into stages, which I think has been very valuable for both conceptual viewpoints about the disease, as well as understanding risk factors and other possibilities for how to intervene clinically, as well as in clinical trial for research studies. So we could potentially argue that this would be stage 0, where there's complex genetic and environmental factors that essentially drive disease risk. And stage 1 essentially has been defined as 2 or more autoantibodies, but the individual still has normal glycemic control. Stage 2 has been defined as 2 or more autoantibodies, but now there's some evidence of dysglycemia, and this could be alterations in an oral glucose tolerance test, but not glycemia levels that rise to clinical outcomes. Whereas stage 3 is now transitioned to the point where polyuria, polydipsia, risk for diabetic ketoacidosis is present, and exogenous insulin is now required to control those symptoms and to keep blood glucose in a normal range. Now, it's at this point that some individuals, but not all, will experience a partial remission. Okay, partial remission is heterogeneous, as we mentioned earlier, in that not everybody will have one. And those that do have a partial remission, the time period for that is highly variable. And as far as we know, largely unpredictable. What I did want to remind myself to say is that throughout all of these stages, autoimmune events are occurring. And I put some crude pictures of immune cells up here in cartoon form, and this will represent a macrophage and a T cell. There's a lot of immune cells that interplay here. We're just showing 2 of these as examples. But it's really just to remind me to say that immune cells are interacting amongst themselves. And then there's an immune cell beta cell interaction that is essentially eventually driving this loss in the ability of the cell to either secrete insulin or in the total number of insulin positive cells, or both, and that this is certainly heterogeneous across individuals. The other thing I wanted to do is point out, and Dr. Wasserfall will talk more about autoantibodies towards the end of the talk, but I wanted to mention autoantibodies in terms of risk for prediction and go back to some seminal data by George Eisenbarth and colleagues that I think is still very representative of what we know today. And what I wanted to focus everyone on is the detection of autoantibodies here. So this triplet has no autoantibodies. And so this was a sort of a rare opportunity for them. They had triplets, one of which had established diabetes, one of which had no autoantibodies. And the other triplet, the third triplet, was autoantibody positive, but at the time they started this study did not have diabetes. All right. So I want to contrast what I'm going to call triplet 3 here with triplet 2. When they measured insulin output in response to intravenous glucose, what you can see is the output for the triplet that did not have any autoantibody detection was much more than that than the triplet that was positive for autoantibodies. All right. And what you can see is over time, this ability to secrete insulin in response to glucose deteriorated to the point where eventually there was an onset of type 1 diabetes, what we would now call stage 3. And this individual, at least across the time point studied here, you can see the one triplet was diagnosed in 1974, and this individual was carried out at least to 1982 and never was positive for antibodies or didn't progress. So in this case, the presence of the autoantibodies predicted the eventual development of T1D. I think that has been borne out in many other studies. And I think now we could probably agree that the more autoantibodies you test positive for, the higher your risk is going to be. The other thing in terms of heterogeneity that I wanted to mention is the immune cell recruitment into pancreatic tissue. This has been one of the things that I think has been somewhat controversial in what is defined as encephalitis or immune cell recruitment into pancreatic tissue, and particularly surrounding pancreatic islets. And this is a study almost 10 years ago now, but I think it's really a seminal paper. Dr. Wasserfall was involved in this study. And it shows in a single patient, you're looking at glucagon in red. So glucagon is really kind of marking the islet, if you will. And CD3 is standing for all of the T cell populations. So CD4, CD8s, Tregs all fall under that CD3 heading. What you can see is in one individual, a single individual, a little bit of immune cells present near this islet, maybe some moderate amount periencephalitis near this islet, and a pretty severe surrounding the islet. And this is all from one single donor. So you can imagine if you got 100 different donors, the heterogeneity that might exist in terms of immune cell presence near and within islets that would be contributors to that disease. So I wanted to point that out because I think it's a key feature of the disease is that heterogeneity and a variety of variables, and that includes immune cell recruitment. All right, so the thing that we also wanted to talk about that I think is at the heart of the discussion today is the metabolic events that might define partial remission. And so for that, we started to think about how is partial remission defined? And again, we wanted to be clear that not everyone experiences a partial remission or a honeymoon period. And those that do, the length can be variable. And part of the, I'll say, problem in the field is that there's no standardized definition of partial remission. And so what we decided to do is just give a few examples of how partial remission has been defined currently. It doesn't mean any of these are right or that we favor any of these or that we're suggesting one is better than another. We're not giving an exhaustive list either. There are more than what we're listing here. But just for examples, C-peptide levels. And so the C-peptide is secreted in a 1-to-1 ratio with insulin. And so stimulated C-peptide greater than 300 picomole per liter is essentially a marker of insulin output by the residual beta cells that remain. Hemoglobin A1C levels less than 7% has been noted as one possibility to denote partial remission. Certainly the insulin dose. So insulin is, the critical therapy for this disease. And exogenous or injected insulin requirement of less than or equal to 0.5 units per kg body weight per day has been suggested as a possibility for partial remission or has been used in that capacity. And then there's an insulin dose-adjusted hemoglobin A1C. And this combines hemoglobin A1C levels, which are essentially a measure of hyperglycemia or glycemia changes over a long period of time, with insulin dosage, which is more of a day-to-day parameter to provide, in theory, more information on glycemic control. Again, all of these have been used to define partial remission in the literature, and there are others that you could find in addition to what we've listed here. All right, so one of the things that we started to discuss among ourselves that really, I think, drove the concept for this article was how does partial remission potentially impact immune cell function? And so this is really the global overview that I'll start with, and then we'll go through each individual cell type. And the concept is that prolonged hyperglycemia, so long-standing elevations in blood glucose prior to initiating an insulin therapy, provides excess substrate for immune cells such as macrophages and T cells. It also places an incredible strain on beta cells. Beta cells like to deal with glucose rises in a pulsatile manner so that they can reset, secrete a normal amount of insulin in response to glucose. That blood glucose lowers, and they have these pulsatile events, which allows them to maintain insulin output to support a physiological blood glucose level. When they're forced to deal with constant prolonged hyperglycemia, this essentially suppresses their ability to secrete insulin, starts to promote losses and changes of maturity markers, and in response to cytokine stimulation from a variety of immune cells, promotes them to make molecules such as chemokines that recruit additional immune cells and may be one of the drivers for that insulitis that we pointed out as heterogeneous among tissue donors and including even just a single donor. And so the idea then is once you are given insulin to regulate that blood glucose into a normal healthy range, that reduces substrate availability for the immune cells that would be within that tissue that will be targeting that beta cell population. In addition, it allows the residual beta cells to essentially recover, perhaps reset, and for perhaps a short period of time, function somewhat normally. And this may be, we think, a contributor to why some individuals will experience a partial remission after they've been given an insulin therapy. This also may be one of the reasons why some of the exciting drugs that we've seen be successful in clinical trials, like baricitinib and teplizumab, extend that partial remission period, because they're now then capable of suppressing some of these cytokine signaling pathways, as well as modulate some of that T cell effector function during a period where an individual might be experiencing a partial remission. So what I was going to do next is just kind of blow up the beta cell a little bit, show you perhaps in just a little bit more detail. This is a picture of a mouse beta cell stained for insulin in green. So you can see this is what a healthy islet looks like, lots of insulin. Beta cells experiencing chronic hyperglycemia display some toxicity, some of which leads to cell death when they have to deal with large amounts of glucose. And when we say large, we mean typically greater than 126 mg per deciliter, whereas the normal range would be 70 to 100. But we also wanted to note, again, along the concept of heterogeneity, that not all of us necessarily start with the same number of beta cells. So if Dr. Wasserfall has twice as many beta cells as I have, and both of us are experiencing chronic hyperglycemia, I might be at a greater risk for toxicity, assuming that we have equal levels of cell death in response to that. The person with more beta cells perhaps can tolerate that for a longer period of time than a person with less beta cells. In addition, the mechanisms that beta cells use to secrete insulin, some of those calcium dynamics, oxidative stress, all of these are altered when they have to see glucose all the time rather than in a pulsatile fashion. In addition, we mentioned earlier that beta cells use glycolysis and TCA cycle to metabolize glucose to drive insulin secretion. But as they deal with larger amounts of glucose, they have to route some of those substrates into other pathways. One of which is the hexosamine biosynthetic pathway will produce hexosamines that modify proteins. And excess hexosamine production in beta cells has been linked to reduced insulin secretion. In addition, forcing beta cells to experience chronic hyperglycemia can promote dedifferentiation, loss of maturity markers. And if I were to use kind of a silly analogy for here, it would be like a person that can tie their shoes no longer can tie their shoes, or you used to could drive a car, now you can no longer drive a car. Beta cells that see chronic hyperglycemia, they no longer are good at secreting insulin. They lose some of the markers that make them a hormone-secreting cell. And in some cases, we think there's been some good immunohistochemistry studies suggesting they may gain antigen presentation machinery expressed. Does that mean they can present antigen to immune cells? Maybe, but I don't know that the evidence for that's great. So you're sort of forcing them to go from a very glucose responsive, very good cell at secreting insulin to a cell that's either dying or dysfunctional, or some populations are dying and some populations are dysfunctional, because mass and function don't always correlate well across this disease. So chronic hyperglycemia for insulin producing cells is not a good thing. And so that's why we think restoring blood glucose with insulin therapy may dampen some of these pathways, allow some of those beta cells that are still alive to recover, reset, restock their shelves with insulin, and potentially be responsive to a glucose stimulus, at least on some small level, for a short period of time. And that may be one reason why partial remissions are very heterogeneous, that individuals can have a partial remission of 3 months or 6 months or 3 years, and you don't know how long someone's going to be in a partial remission if they experience one at all. All right, so if we go back to our 30,000 foot view and switch gears a little bit to the T cell, one of the things that intrigued us is that T cells are a metabolically driven cell, and when they go from a naive state to an activated state, they ramp up the amount of glucose metabolism they use. And I'm particularly talking about effector T cells here and not so much Tregs. All right, so in a 70 to 100 mg per deciliter range, so this would be what we would consider a normal blood glucose, the amount that is sufficient to drive processes. And we're going to use interferon gamma as just an example, because the primary data, if anybody's interested in the original papers, are listed at the bottom, studied interferon gamma as the primary outcome. And so what we sort of propose is that as blood glucose rises, this promotes the availability of excess substrates for the whole T effector population that might be present in pancreatic tissue or within islets or in pancreatic lymph node, to essentially increase the amount of interferon gamma transcription. We know this is coupled to glucose. The amount of interferon gamma transcript that is loaded on the ribosomes is coupled to glucose metabolism. And that tracks with the amount of interferon gamma secretion out of the cell. Beta cells are responsive to this particular cytokine, can promote activation of transcription factors, telling the beta cell to make molecules that recruit additional immune cells. right? So one of the things I wanted to point out for the T cell is that mitochondrial metabolism is not required to support interferon gamma production. It is absolutely required for them to undergo glycolysis. For T cells to proliferate, they can use either one, glycolysis, mitochondrial metabolism, but to support interferon gamma production, they absolutely require glycolysis, but not mitochondrial metabolism. And the sort of idea that we put forth was that lowering blood glucose with insulin therapy would essentially take away some of this substrate availability and potentially, we don't know for sure, this is just an idea, reduce the amount of cytokine that would be available for driving autoimmune events. Switching over to the macrophage, the concept is somewhat similar in that, except that mitochondrial metabolism is absolutely necessary to support IL-1 beta production in the macrophage. This is different than what we saw with interferon gamma production from the T cell. Again, if anybody wants to see the original data, it's from Luke O'Neill's group, published in Nature. But the idea essentially is if you have a normal blood glucose range, there's going to be a finite supply of glucose to support metabolic activities in the macrophage. After priming, if you happen to be in a window of elevated blood glucose, this would provide excess substrates. And what the macrophage does upon activation is use glycolysis, TCA cycle, and TCA cycle substrates to inhibit an enzyme. So this is a prolyl hydroxylase enzyme that gets inhibited by succinate. That stabilizes a normally unstable transcription factor. And the stabilization of this transcription factor allows the macrophage to produce increased amounts of cytokine. And so I'm showing interleukin-1 beta as an example here because that's what Luke O'Neill's group did in their original paper. And also because beta cells are highly responsive to IL-1 beta in terms of chemokine production. Now, we're not saying that we think interferon gamma or interleukin-1 beta are the panacea for T1D development or progression. We think they're examples of inflammatory cytokines made by immune cells and very likely seen by beta cells during the course of the disease. We also are proposing the possibility that lowering that blood glucose from a pathologic level back into a normal range may decrease the amount of substrate available for those immune cells targeting beta cells, ostensibly leading to less inflammatory milieu in the pancreas. And so what we propose is that possibly slows down immune cell recruitment and may help slow down disease progression. All right, at this point, I'm going to turn it over to Dr. Wasserfall so he can correct anything I've said thus far or talk about the really interesting autoantibody production component that he wanted to mention today.
Okay, thank you, Dr. Collier. So next slide, please. This is an extension of the paper. This wasn't part of the paper, but thank you, Dr. Wesley, for inviting us because it prompted me to think a little more deeply about how much immune stuff we actually study after the onset of diabetes. And so the autoantibody story is mostly a pre-diabetes and diagnosis marker. We don't typically study a lot of them in long duration unless we're trying to sort out type 1 from type 2. But what we do know is that it's not as reliable after the onset. And this is because if you look at this graph, y-axis is the autoantibody titers. The x-axis is duration of diabetes with 0 being diagnosis date. And we know, and we've known for a while, that GADS tends to hang around. I2 and ZNT8, as you can see, wanes after onset. And this has mostly been interpreted as sort of loss of antigen, so equating to loss of beta cells. GAD is found in other sites in the body, so maybe that's why it continues, and it may even be in alpha cells that may keep the immune system stimulated a little bit. But this also occurs in thyroid disease. If you actually remove somebody's thyroid when they have autoimmune thyroid disease, the antibodies go away. So this is an antigen-specific loss that drives down the autoantibodies. And that segues to the next slide, please. Here we see that if you give the antigen back in the form of a transplant, the antibody titers go back up. This is a recurrence of autoimmunity rather than an allo response, because my GAD and my ID2 is the same as anybody else's. There's a little bit of variation in ZNT8, which is not shown on the slide, but that's fairly polymorphic, so you could have an allo response there. But most of the allo response will be generated towards mismatched MHC in the transplant. This is indicative. This looks like a boost response. You give an antigen back and the antibodies go back. So during that remission phase, which is shorter than the loss of antibodies, the antibodies are starting to wane, and the eventual loss of them kind of implies that the antigen is all gone. So next slide, please. If you look at this slide, this is a short duration, 3 years of duration. It's from the NPOD collection. This person had all 4 antibodies, autoantibodies. And here you can see in one view all the different kinds of islets. Well, not all. 4 of the main kinds of islets you would see in type 1. So you have an insulin positive islet, insulin in red, along with CD3 staining showing insulitis. You have what are called pseudoatrophic islets. These are islets that have lost insulin and they're only glucagon positive and they do not have CD3 infiltration. And then there's an insulin positive islet that does not have insulitis. So we don't quite understand why the islet on the right of this view is being attacked and the one on the left is not. And the ones in the middle have already lost their insulin and the immune response has kind of gone quiescent, which kind of sums all of that up together. Once the entire pancreas becomes insulin negative, you may not have antigen left, the antibodies go away, but you still have memory. So if you have a transplant or if you have a stem cell transplant, you are going to get a reemergence of an immune response. And so in the next slide, I just wanted to kind of bring this together. And this by no means that the triangles, don't think of them as absolute high and low. It's just that as we move across different kinds of diabetes, and type 1 is the one we're talking about, is that the inflammation component changes across the spectrum of diabetes, but also during the course of type 1. So how much innate immunity is required to trigger an immune response is understood in normal viral and bacterial infection disease, less understood as to how it triggers autoimmunity. The specificity of an antigen-specific adaptive immune response you can see in type 1 diabetes and maybe some checkpoint inhibitor cancer therapies where you induce diabetes as a side effect. The actual implies that there's a memory component to it. If you move to the left, there isn't a memory component to it and it's inflammation. But similarly, if glucose is high, you're going to exacerbate that inflammation. If glucose is high, you're going to exacerbate the T cell response. And so I think my take-home message was we have to deal with inflammation. We have to deal with a specific immune response. We have to deal with beta cell health. And we have to deal with metabolism. And to get to the true therapy, we may have to manage all 4 of those aspects of this disease. And so I'm going to hand it back to Dr. Collier to summarize for us.
Thank you, Dr. Wasserfall. That was a great summary. And I agree. I think where Clive was going with that is we're definitely going to need combination therapies and precision medicine approaches. And, he and I both agree with that. And I think that's probably going to be shared with some people in the field at this point and hopefully moving forward as well. I think finally, I just wanted to say that most of us agree, if not all of us, that T1D is a complex disease, both genetic and environmental factors, heterogeneity among patients, and even if we're talking about immune cell presence, that is insulitis within islets, there's heterogeneity even among the same donor. So basically, we presented an idea, and I'm very clear that this is an idea. Clive and I are not saying that these are definitive facts, but the idea is that substrates needed to enhance immune cell function can be reduced by lowering blood glucose into a normal range. We think that the substrate overload outcomes that are negative for pancreatic beta cells can be mitigated by lowering glucose levels with insulin therapy. The link between blood glucose control and modulation of immune cell function, as Dr. Wasserfall said on the last slide, are really critical components, we think, to understanding and suppressing these autoimmune events that essentially lead to loss of insulin availability. And that the partial remission period, it appears to be extended by specific interventions in these discrete patient groups. Particularly, we're thinking of baricitinib and teplizumab, which have been really exciting in the last 2 years. And just to kind of sum up, a lot of that initial JAK inhibitor and anti-CD3 therapies were borne out by beautiful studies, preclinical studies using the NOD mouse that were both prevention, both anti-CD3 as well as the JAK1/2 inhibitors did well in reversal studies in the preclinical models. And this so far, we think has translated pretty well to some of the recent clinical trial outcomes. And so we're really excited about, combination therapies, precision medicine approaches, and roughly the idea that is lowering blood glucose into a normal range, possibly also immunomodulatory. And that's essentially what we wanted people to take home today. And we'll stop here and acknowledge people that have helped this discussion. Dr. Daniel Shaw is a pediatric endocrinologist at Emory. I bounced some of these ideas around with him, especially just in terms of how does he define partial remission in his pediatric patients. Mike Brehm and Mike Karlstad were co-authors on the article we presented today. Dr. Susan Burke is another allobiologist at the Pennington that has been subjected to my discussions of these concepts. And probably most importantly, families and patients, the people who deal with this every day, the trial participants, the organ donors are absolutely essential to everything we do. And so we wanted to give a big thanks to those folks. And then if anybody would like to contact me or Dr. Wasserfall, our emails are here. We're more than happy to hear additional comments, questions. If you have a reference you think we've missed, we would love to hear from you. So we'll stop here and take any questions if there are any. Thank you, everybody. And thanks, Monica.
Yeah, thank you. Great talk, and really diving into some of the sort of darker corners that haven't really been discussed that much in the literature up until you put this paper out. I mean, recently. So yeah, open it up for questions.
Hi, my name is Benjamin Watson at the Th1nk Children's Medical Center in New York. Thank you for the wonderful presentation. I learned a lot, and this is the field that everybody is trying to navigate and figure out what's going on. So my research in this area is based on vitamin D. So I have to make a few comments on your approach, which is maintenance of normal glycemia after stage 3, with the hope that it will decrease autoimmune, activity and then prolong the partial clinical remission phase. Now, as you know, going back to DCCT trial, it did show that if you maintain glycemia, you could actually prolong the partial remission. However, since then, there are a lot of studies that have actually not borne out that same conclusion. One I will cite right now is the study by Forlenza, which was published 2 years ago in JAMA, where they studied verapamil and partial remission. They did show that there was some significant slowing of the decline in AUCC peptide, but they did not find any impact from maintaining blood sugar. And they use CGM, they use AID techniques and everything. So that's just one. I mean, there are others that also show that, normal glycemia would prolong. So that hasn't been settled really, especially recently. Then the next thing is this teplizumab. I know you mentioned it a few times, and the impact on partial remission, meaning after stage 3. As far as I know, based on the available studies, yes, teplizumab prolongs stage 2 to stage 3 by 2 to 3 years, maybe sometimes 5 years. But it hasn't really been very, very strong in terms of what happens after stage 3 is reached. It did show, again, some significant slowing of the AUCC peptide, but it did not have any metabolic outcome that was really meaningful. It didn't even reduce insulin requirements or A1C. Even the meta-analysis they did not show anything. So that's the other thing. Finally, as long as you are looking at normal glycemia, I was wondering whether you're also looking at lipid parameters, because I have done some studies looking at lipids after the diagnosis of type 1, showing that those who underwent remission had actually significantly better lipid profile than those who did not, meaning that the long-term complications of diabetes, which partial remission could help remit or reduce, is not just a function of glycemia, it's also a function of lipid changes. So those are my comments. Thank you again, both of you, for the wonderful talk.
Thank you, Dr. Wasu, and I apologize if I mispronounced your name.
No, it's good.
Okay. I've read some of your papers, so thank you for joining. It's nice to see you in person. Your points are well made, and I think some of these concepts, and Clive can jump in to add or correct, but probably due to, differences in trial design, differences in endpoints, certainly differences as to way partial remission is defined. But I think you're absolutely right on with the drug studies. The second piece, your lipid comments, I think are incredibly intriguing. Okay. There's a great article by the late Dennis McGarry that talks about what if we defined diabetes by lipid rather than by glucose. Okay. And he was referring more to type 2. But I think the concepts, especially with you bringing up the points here, may also be applicable to type 1 because, when we lose insulin, we're not just affecting glycemia, we are affecting ability to metabolize lipid normally, right? I think your points about measuring lipid or how do lipids play a role are absolutely valid and need certainly more digging deeper into that aspect of it. So I appreciate that comment.
Thank you.
Yeah,
And
I would also add that we've just had a couple of talks recently that really dug into that idea of the lipid profile being very important in the onset of diabetes, and that, Karsten Bouchard as well as Th1nk Wan from WashU, both were kind of bringing that up. So important point. Thanks for bringing it up.
Sure.
And I would just add in with the idea of the teplizumab trials and things like that. I think there is certainly heterogeneity amongst everybody with type 1, but one of the big ones is age. And younger individuals have a faster onset and a less slow trajectory just by definition, really. And I think their honeymoons may be a little bit different too. But I think the concepts are, what we're hoping is the concepts of we're going to have to treat a lot of different things, not just insulin replacement.
Has anyone done a standardized honeymoon evaluation between those who get type 1, 5 and under, and those who get it later in teen years and then as adults?
I think the new studies I'm aware of is the rapid loss of C-peptide in those under 5 compared to older. The immune response is somewhat similar. Sorry.
No, you're right. The studies have shown that if you're less than 5 years of age, you have a fulminant endotype of type 1 diabetes. So you lose your beta cell mass faster. And there was a study we did in 2017 showing that if you, at the time of diagnosis, if somebody has 4 parameters, meaning younger age, 3 or more autoantibodies, aka or low bicarbonate, they are less likely to go into remission. And the predictive value was 70% if they have those 4 parameters. So younger people are less lucky, let me put it that way, than the older children.
I wonder if an idea is, what Dr. Collier showed the old George Eisenbarth, where the insulin production was already down prior to the onset, whether lowering sugar, maybe not even with insulin, but just lowering blood glucose pre-onset in high-risk individuals would be worth trying as a component.
Has any study been done where insulin was given in stage 2 or even as far back as stage 1?
So the DPT used the subcutaneous insulin at a dose that would not alter significantly blood glucose. It was used as an antigen to tolerize, not as a metabolic agent.
Right.
However, if you want to extrapolate, there was that study everybody knows, the oral insulin trial by TrioNet group, multi-center and everything. Everybody thought that was going to answer the question, but it did not. Now, somebody can, of course, question oral insulin. Was the insulin destroyed by, stomach enzymes and acids and all that? We don't know, but, the outcome was really non-significant.
Anna Kazu here, Advent Health, Orlando. Hi, Clive.
Hi.
I have a question about the glucose levels. At the beginning, you showed a beautiful islet studies with differences in insulin secretion with as little as 10 to 20 milligrams per deciliter going from 60 to 80. My question is, would that apply to the immune cells? Like the immune cells will have an effect for these small differences in glucose. And if that is the reason that something that is happening, when we maintain patients in euglycemia, quote unquote, we are not close to 80 milligrams per deciliter. So maybe the failure of all the studies that have been done in early metabolic compensation is simply because we are already above the target. Any comment on this?
I mean, I think right now that's reasonable. You know, again, you know, I think Clive would agree. What we're presenting is an idea. We don't have definitive data to answer that question. But what we're hoping is, you know, you come and Dr. Wasu coming in with interesting comments and thoughts that can help not just Clive and I refine studies, but if anybody else is intrigued enough to have any of these concepts help their own study designs, we would love to be able to answer those types of questions. Right now, I just don't think there's data for it. Clive, did you have something you wanted to
No, I
add?
was just going to say the scary part of that is it's worse to induce hypoglycemia than it is.
No, I guess a
Clinically, yeah.
question is how long do you need to keep that metabolic compensation to have the cell recover, and maybe you can do it again. We can maintain safely those levels when we do clamps, but
Right.
how long can we clamp a person with an IV
Yes,
infusion?
exactly.
Or if, I don't know, dual hormone pumps
kind of an-
will help.
Kind of an interesting philosophical question there is,
Right.
we may have actually had some successful immunotherapies, but we don't know because the beta cells continued to deteriorate. Even though immune pressure was off, it was too late. We may have had some successful beta cell therapies, but we never dealt with the immune response. And the transplant ideas kind of show that you're going to get recurrent autoimmunity and allo rejection. So I think we haven't tackled everything at the same time. We may have masked some successes.
Can I bring in sort of this question of data to both of you? You know, Dexcom obviously has a huge cache of data, Tide Pool, et cetera. What about, you know, looking back over those who were stage 0 at high risk genetically, stage 1, stage 2, looking back into these data sets and seeing whether or not you can kind of identify relapsing, remitting patterns, if you could identify, you know, and connect to who stayed in remission or honeymoon longer once diagnosed, things like that. Is anyone interested in or doing that now?
I think, I mean, that sounds like a retrospective secondary data analysis. I'm not aware that that's been done. I personally think it would be interesting. It might be one, I don't want to say simple, but one quicker way rather than, you know, as opposed to designing a new study to try and
Just to give
test
some
the hypothesis.
guideposts. Yeah.
Yeah, I think it's a great idea. Clive, are you aware of anybody that's done that?
So Ezio Bonifacio has published within, I think it's maybe, I don't know if it was Teddy or his own baby diet or the diet one in Germany. I can't remember which one he reported, but anyway, where they actually had been measuring this, and glucose elevations occurred prior to the first seroconversion to an autoantibody.
Yeah, I mean, that paper's been out, what, about 3 years now. I think Annette Ziegler's on it.
Yes.
Yeah, I mean, that's a compelling, you know, idea that some of the hyperglycemia for some individuals pops before the autoantibody detection.
And then you could kind of start thinking about binning individuals by the length of their so-called remission, you know, states.
Right.
Interesting idea. I had another question about the hexosamine production too. Is that detected in blood? Could it be used in staging, or is it just too minute?
Yeah, I mean, I don't know. All right, but I view that as an intracellular change that is associated with modifying proteins. It would probably require being able to detect perhaps either a cell membrane, so a cell surface marker that's been modified that way, and whether or not there's an increase or decrease. That might exist, but I don't personally know what it is.
Yeah, I just wonder.
Yeah.
If the cells are sort of blowing up, you know, is it freed up into the bloodstream? But yeah, I guess my last question is, how would you both, sort of in an ideal world, design blood or imaging biomarkers to predict honeymoon duration or therapeutic response? What would be sort of some first steps you would be thinking about?
That's some rich bait. That feels like a conversation that has to be continued at the bar.
this could be a whole nother talk.
Yeah, I think, yeah, there's lots of variables to consider. I don't think there's any one definitive thing I could say today that would answer that. Claude, do you have anything you
Right, and
wanted
I
to?
think that sort of measuring everything all the time is a tough thing. They've done Th1nk in the Teddy, they've done mass spec, they've done lipidomics, all those kinds of studies, but it's probably not minute to minute or day to day.
Right.
So there's some intriguing things like in the TNF pathway, which goes along with the macrophage type production of inflammation. Whether those markers will be sensitive enough to show relapsing remitting. I mean, even in rheumatoid arthritis, the relapsing remitting is mostly about joint pain rather than actual biomarkers. We just don't have, or lupus, whether you have the butterfly rash or not, or skin lesions or not. Vitiligo, whether you see patches clearing or not. We have nothing at this point that shows beta cells coming and going.
Yeah,
Oh, you said
yeah,
something,
exactly.
though. Sorry, are you saying something, Dr. Collier?
Oh, I was just going to agree with Clive and just saying, more frequent longitudinal data would probably be needed. But I think Monica's question is more, what do you measure all the time in a perfect world? And I think there's lots of things.
What would you start with? Yeah, what would be your first pick, basically?
So let me say two things. Number one is in Boston, they have this thing called the medalist study. So if you have diabetes for 50 years, they give you a medal. So they follow those people with serial C-peptide study, other studies, and also some of them sign, to give away their organ when they're dead so they could stain the pancreas for, beta cells. So they have really used very rigorous techniques to show that some of these lucky type 1 diabetes patients who are microsecretors go on to continue to make some insulin. And so that is really the perfect scenario for somebody with type 1 diabetes. So they track C-peptide. I believe they also use insulin dose adjusted A1C values to show that these people have continued to make insulin. Then the other thing I wanted to add is that I know, Dr. Collier, you probably, this thing called theory of hyperglycemic memory, which they believe that the glucose excursion at the time of diagnosis of type 1 diabetes at the stage 3, right, will determine whether you will even go into remission or whether you have long-term complications. The body remembers all these things. However, as I was coming up the rank, I was always struggling with this thing because it's an incomplete theory. You cannot just focus on sugar only. You have to look at lipids. So after looking at lipids in children over 5 years and also looking at lipids in adults with newly diagnosed, I came up with the fact that you have to match hyperglycemia to hyperlipidemia and find what's common with these two conditions in a subset of people with type 1 that would then go on to explain the higher prevalence of microvascular, so glycemia, and macrovascular complications, that's lipid, in this population. I believe that you must join lipotoxicity with glucotoxicity to get good answers about complications.
Yeah, I think those are good points. And especially when, insulin drops and glucose rises, and then you're forced to start using lipid stores. So you get increased lipolysis, right? So you get increased blood lipids. I've always been somewhat intrigued by the idea that certainly the lipid's going to be taken up and metabolized, but some of those lipids may be stimulators of pattern recognition receptors that support this inflammatory response. Right. And so that may be one of the reasons that lipids are so important, right? Not just, as a fuel source, which they certainly are important for that, but as potential triggers of inflammatory pathway signaling.
Absolutely.
All right. We're at the top of the hour. This was such a great and rich conversation and discussion. Really appreciate everyone's contributions and expertise. And we look forward to hearing sort of next steps. It sounds like perhaps a collaboration is brewing here.
Yeah.
Always.
And that's great. And so have a great rest of your day. Be careful riding out the storms there in the south, and have a great rest of your day, everyone. Bye-bye now.
Thank you.
Thanks, Monica. Thanks, everybody.
again.